I would like to welcome the final speaker for the session. Alvaro studied biochemistry, obtaining a PhD in biomedical sciences at Universidad de Chile, obtaining a PhD in 2007. He then continued his postdoctoral training at the Cancer Center Karolinska at the Karolinska Institute, Sweden, working on T cell-based cancer vaccines with Dr. Rolf Kessling. In 2010, Alvaro started a career as an independent scientist at the Fundación Ciencia y Vida. Since then, Alvaro has developed his research focused on unveiling the cellular and molecular mechanisms underlying effective and long-lasting T-cell mediated anti-tumor immunity. His current projects aim to decipher the crosstalk between CD8 positive T cells and dendritic cells in the tumor microenvironment, and to dissect the role of tissue resident memory T cells, or TRM cells, in human solid cancers. Welcome, Alvaro. We are excited to hear your talk.
Hello. Thank you very much for the invitation and the opportunity to present and share our data. This is an ongoing project. So I will give a brief introduction on the topic. So we're interested on tumor-specific T-cell responses, which are initiated by antigen-presenting cells, such as dendritic cells, which present antigens derived from the tumor to T-cells in the lymph nodes. And all the T-cells, they bear TCRs, or T-cell receptors specific for those antigens, are amplified, are expanded, and they differentiate into effector T-cells. These effector T-cells can then go migrate to the tumor site and then recognize tumors or antigens presented by tumor cells and eliminate these cells by cytotoxic mechanism, including the release of granzyme B and perforin. So this is how the responses are initiated, and then in the time we have these cells that expanded. Most of them are reduced in the contraction phase, and only the memory phase is generated when these memory precursors become memory T cells. And when we talk about memory T cells, we can divide these cells into big subgroups. So the circulating memory and the tissue resident memory. So we're interested in the tissue resident memory T cells because they become established in barrier and non-barrier tissues, and they have a long lead, and they acquire a distinct residency signature, including the upregulation of different markers such as CD69, CD49A, and CD103. These molecules allow them to interact with the different epithelial cells and also parenchymal cells. So why are these cells important? Is because they have a potent effector function. So they produce large amounts of gamma interferon, TNF alpha, IL-2, and cytotoxic molecules such as granzyme B and perforins. But they also orchestrate the response because they trigger an innate-like alert state in the tissues. They become to the recruitment and activation of different cells, such as dendritic cells, other circulating T cells, and also NK cells. So this is the cells that can orchestrate the response. And in mouse model, it's been known for a few years now, and that these cells can become very important to orchestrate the response. Yeah. So some of the groups from Spain and US, also some of our own contributions, have allowed to demonstrate that the tissue resident and circulating T cells act synergistically to reduce the tumor burden and to have a better response against tumors. But this is all in a mouse model. So in humans, we also have done some analysis using the databases from the Cancer Genome Atlas. And we can see that the patients, they have enrichment of TRM cells, they have better survival in this type of analysis. Also in multivariate analysis, this is a one of the factors that allows the patient to live longer. Okay, so this is the background, and this is not only true in melanoma, but also in other cancers like breast cancer, lung cancer, and so on. This is how these cells, which are here represented in yellow, they insert in the tumor isolate. And they, of course, they produce a better or a longer survival in the patients that have these cells compared with patients that have only circulating T cells, so they do not have any infiltration of CD8 T cells. So this is what we got interested in this cell. And our main questions at this moment is, what is the tumor reactivity of these cells in human cancers? But also, what is the phenotypic and functional heterogeneity of these cells in different types of solid tumors? And for this, we have done some work at this moment in renal cell carcinoma tumors. As you can see here, we're isolating cells from the renal cell carcinoma tumors, but also in some samples from healthy tissues. And as you can see, we can see that at least two types of TRM cells which express the C69 rich tissue resident marker. The ones that do not express CD103 and the ones that express both markers. So we call this the CD103 positive and CD103 negative cell. So this is the quantification, and this is as a control, we use cells analyzed in the blood. As you can see here, these cells are quite enriched in tumors, as you can see here, as compared with circulating cells. So we have done some phenotyping of these cells, and we can see that, for instance, the double positive, they express this canonical tissue resident marker RLX3, they express higher level of PD-1, and also they express the CD49A, which is integrin that allows to interact with the collagen. So this is the quantification of the cell expressing these markers between the non-TRM cells, the single positive and the double positive cells, as you can see here. So there is expression of this canonical marker. We have analyzed some other markers, but I won't show all of them. So one of the questions is if these cells can recognize tumor cells. So for this, we can culture, we have developed a system to culture the non-tumor renal tissue and also the renal cell carcinoma cells that we can expand in vitro for the 3 weeks. We also isolate the tumor-infiltrated lymphocytes, and we can cell sort the non-TRM, the single positive and the double positive cells, and then co-culture this cell in co-culture studies. And to show the dependence of the recognition, we can also block the MHC class 1 molecules to avoid the interaction of these cells, or use a positive control like an agonistic anti-CD3 antibody. In this figure, I can just want to summarize that these cells, when they are exposed to tumor cells, they upregulate 4-1BB, meaning that they are being the antigens through the T cell receptors. When you block these HLA molecules, these activations measured as 4-1BB upregulation is decreased to control levels, and these cells cannot recognize normal renal tissue cell. So this is showing that these cells can recognize autologous renal cell carcinoma cell. And this is the quantification for the 2 types of TRM cells we're working on at this moment. So we have quite around 50% of the cells that are in the tumors can recognize the tumor cells, but not the healthy tissue. Something interesting is that it seems the double positive cells, they produce less gamma interferon when they are activated with the tumor cells or when they are activated with agonistic antibodies, meaning that these single positive cells have a better functionality. So then to analyze the expression of the different markers and the heterogeneity of these cells, we have performed single cell RNA sequencing using the 10X platform. And we are analyzing at this moment the transcriptome, the TCR profiling, and also we can measure the cell surface protein using featured barcode antibodies. At this moment, I will only focus on this presentation, I will only focus on the transcriptome. So using this, analyzing only memory CD8 T cells infiltrating renal cell carcinoma tumors, we can identify different clusters of cells that are defined by differential gene expression analysis. Okay, so these are around 10 subsets of memory CD8 T cells that we can find in the tumors. If we do a UMAP analysis, we can see that there are different of these cells, and some of them are considered TRM cells, like the cluster we can see here. This we can call exhausted TRM cell because they have many inhibitory receptors. We have other group of exhausted TRM cells, and we also have a cluster that we call TRM CD103 positive cells. We also find some cells that are in cycling and some other that have other features like the highly cytotoxic TEMRA cells, TC17-like cells, and effector memory cells infiltrating the tumors. When we analyze these different clusters, we observe that they have some genes in common, but also some important genes that are exclusively expressed in one of these different TRM clusters. Importantly, is the CD103, the CD69, and the CD49A, which we analyze by flow cytometry. When we analyze also this, we try to compare what is the transcriptome of this cell as compared with previously reported TRM cell gene signatures. So this is a paper in breast cancer where we can see that it's very similar, the expression of the genes that they found as a gene signature for their TRM cell to ours, which are in the cluster 2 and the cluster 8. This is data from single cell RNA sequencing. When you compare with this, in the same work with the signature obtained from the bulk RNA sequencing, this is a little bit different. And we also can see that this is highly enriched genes are in this cluster 4, which is cycling, and probably could be a TRM that are going to proliferation. And this is another study used mainly in mouse models, where we can see that there are upregulation of the genes in this cluster that we call the TRM CD103.
Alvaro,
yes.
This has been a really exciting presentation, but just for time to make sure people have time to ask you questions, would you be able to move towards the conclusion?
Yes.
Thank you so much.
This is just on the one slide ahead.
Perfect.
So, this is a comparison with other exhausted and effector memory signatures that are very important in this case. So, as conclusions, we can say that TRM cells are relatively abundant in RCC tumors, that they can recognize renal cell carcinoma cells, but not normal kidney cells, and that these represent a phenotypically and functional heterogeneous subset of tumor infiltrating lymphocytes. At this moment, we're analyzing the TSL profiling to know what are these cells that are expanded. So just I want to thank especially to the people involved in this study, Sofia, PhD student, and Vincenzo, which is a colleague, MD, PhD. And also the group of Elian Piaggio, which is very instrumental to do this single cell RNA analysis. Thank you.
Thank you so much, Alvaro. Such an action-packed presentation full of a lot of information. So as people have questions coming in for you, we have just a couple of minutes for that. But as we get started, it'd be interesting to hear about, to foster growing collaborations in the scientific community. Could you speak to collaborations around this work that would be exciting to you?
Yes, I mean, at this moment we are collaborating with the different groups, okay, here in the Curie Institute and the University College London, the group of Sergio Quesada. But it will be very interesting to bring this collaboration with more Latin American community. So we're very eager to establish some new collaborations with the people working on this type of projects.
Thank you. I hope those interested would reach out and during and after the meeting. So what do you think next steps are for this kind of work?
So at this moment, something very important is to know these cells, what is the TCR profile of these cells. So to know what are the cells that are being expanded, what of these different TRM cell subsets are being expanded in the tumor, and to know what is the origin. So we are also analyzing at this moment the healthy tissue, the juxtatumor tissue, and we have some interesting results that maybe these cells are becoming from the outside of the tumor, and we have like a pool around the tumor that is that could act as progenitors of these more differentiated cells.
Awesome. Thank you. Well, this will conclude this session. It is really exciting to get to know our session scientists today and hear about their science that is happening in Latin America. And I'm just looking forward to the next session. But for now, time for a break. So if everyone could please come back to us at 3:30 p.m. Eastern Standard Time, 3:30 p.m. Eastern Standard Time. We'll resume then. Thank you so much for attending this session.